Okay, we're on question 20. Pathogens cause disease and are transmitted from individual to individual in a variety of ways. Which of the rows A to D in the table below is correct? So this is disease, so it's my stuff. A, athlete's foot. B, HIV. C, malaria. D, TB. So I would be thinking, before I look at that too much and get confused, I'm thinking, right, athlete's foot, that's fungus. HIV is a virus, malaria is a protist, and TB is bacteria. So let's see, so that matches up, that doesn't, and that doesn't. So they should be the other way around. Means of transmission, well, athlete's foot you can get by someone walking on a particular surface and then you walk on it and get it yourself, so that's indirect, and direct from like rubbing feet, I guess. So direct touch or sharing shoes, being direct and so on. So yeah, that makes sense, indirect and direct. Let's just double check. HIV and AIDS, that's spread by mixing of bodily fluids, so sharing needles, unprotected sex and so on. Indirect contact, no, that's definitely wrong. Vector for malaria is correct. TB, direct contact, well, it's droplet infection, so no. So we are sure that it must be A. Okay, next one. Figure shows some of the apparatus used in an experiment investigating water potential in potato tuber tissue. Okay, so again, this is a bit like a GCSE question. Potato in a sugary solution, sucrose solution in a boiling tube. The discs were placed in boiling tubes containing sucrose solutions of different concentrations for 4 hours. The percentage change of mass was then calculated. So when there was no sugar in the solution, the potato gained mass. Makes sense because water has entered the potato, H2O into potato by osmosis. Here again, water's gone in, here again, water's gone in, here water has come out of the potato. And so on. So the water potential of the potato must be somewhere between these two, because it's going to be at a point where no water is leaving or entering the potato, so there's going to be no change in mass. State 2 details of the procedure that must be followed to obtain valid results. So there's a range of things that you could choose. If you think back, you do this one at GCSE. This one I think is Miss Dixon's side, but do it at GCSE as well. So you could use a core borer. Yeah, core borer, that's the right one. So use the same sized core borer for all of your cylinders of potato, and cut discs the same size. So you've got the same shape here and you've got the same size. Another thing, you need to make sure that you blot the potato before you weigh it, before and after. So blot potato dry before weighing. You could also say the same amount of time, they've sort of already said it, but that could be another one. The same volume of sucrose solution. And you have to say volume, not amount. Same volume of solution in each case. Left the same amount of time they've said. Anything else? Same temperature, I guess.
same surface area of the discs, but we've covered that really with the same size core borer, not cork borer. Next, explain how a student could use the data in the table to determine the water potential of the potato tuber tissue. So I would start by saying, where are we? At a water potential of 0.2 mole per decimeter cubed, at 0.2 the potato gained mass, and from that we know that the water potential of the potato must be, let me just think, must be more negative for it to have drawn the water. Yeah, so the potato gained mass, so the water potential of the potato must be greater than 0.2, and it does say explain, so I'd put must be greater than 0.2 because water has entered the potato by osmosis from a now you can either talk about it in terms of more dilute to more concentrated, or you can talk about it in terms of negativity. It doesn't matter, whichever works best for you. So water has entered the potato by osmosis from a I'm going to say more dilute solution, brackets, less concentrated. This is just for you, you don't have to write all of these things. Less concentrated or less negative, or you could say higher water potential. Any of those things, whichever makes the most sense to you. Okay, because it's closer to 0. So water's going to go from where it's closer to 0 to where it's more negative. So water has entered the potato by osmosis from a more dilute solution to a more concentrated one. And again, you could say more concentrated, which I said, more negative, lower water potential, any of those. You could then say that at 0.3 molar water leaves the potato. So at 0.3 molar per decimeter cubed, water leaves the potato by osmosis. I'm not going to go into it all again because I've already said it once. By osmosis, suggesting that the water potential of the potato is greater, water potential is, if water leaves it, it must be
greater, sorry, is greater than 0.3. Less negative. I've gone a bit wordy there, maybe a bit too wordy. Probably get away with just saying at 0.20 molar per decimeter cubed, it gained mass because water has entered by osmosis. And then, so like the same thing here, probably. And then you need to check the question again. Explain how a student could use the data to determine the water potential. So we've explained why we know it's going to be in between these. Explain how a student could use the data. So they know it's going to be somewhere between 0.2 and 0.3. Water potential must be between 0.20 and 0.30. They could repeat the experiment with what's the word that I want? Not a scale. They could repeat the experiment with smaller increments. Does that make sense? Between 0.2 and 0.30. When the potato does not gain or lose any mass, that is the water potential of the potato. Okay, like I said, I think I've gone into it in a bit, probably a bit too much detail up here, but you could just say at 0.2 molar per decimeter cubed, water enters the potato by osmosis, suggesting that the water potential of the potato is lower than this. Then at 0.3, water leaves the potato by osmosis, suggesting that the water potential is greater. The water potential must be between 0.2 and 0.3. They could repeat the experiment with smaller increments. That should say between 0.20 and 0.30 molar per decimeter cubed. When the potato doesn't gain or lose any mass, that is the water potential of the potato. Identify the parts of the cell labelled X, Y and Z. So like I've done before, I would probably just see if I can label them all first. So you'd automatically think that that's going to be the nucleus, but here's the nucleus. Now it's a potato, so that means that it's a plant, so this must be our vacuole. Vacuole, so this, Z, must be our vacuole membrane. Y will be our cell membrane, or cell surface membrane. X will be our cell wall. V will be our cytoplasm. And then W is the space where the cell membrane has been pulled away from the cell wall. So that's going to be filled with this 0.45 molar per decimeter cubed sucrose solution. So we would have the 0.45 solution in that space there, because that's what's caused it to, I can't think what the word is,
I want to say plasmolyze, but maybe I've made that up. I'm sure you'll tell me. Identify the parts. So X is the cell wall. Y, cell surface membrane, and you do have to say surface. You don't get it if you just put cell membrane. You could say plasma membrane, I think. Just double check, pretty sure you can. Yeah, you can say plasma membrane. You could also say plasmalemma, apparently. Yeah, and then vacuole membrane, or you could have tonoplast for vacuole membrane. So all of this question would be Miss Dixon stuff. What would be found at double in the immersed cell? Now, a lot of students, and you'll see when you have a look at the mark scheme as well yourself, always look at the examiner's comments, because I think it's really useful to know what the common mistakes are. A lot of students put that that's just an empty space or an air space or a gap. There's not going to be nothing there because it doesn't form a vacuum, okay, and it's not just going to be air, it's going to be the solution, okay, so it's that 0.45 molar solution. I think you get away with just saying solution. Yeah, sucrose solution. Okay. You can just hear my cat casually eating some cat biscuits in the corner. Okay. Okay, halophytes or halophytes are plants that have the ability to live in soils with a very low water potential, so very salty areas. In the UK, these plants form part of salt marsh communities. So halophytes, we've heard of hydrophytes. So that means, phyte means to do with living. So hydrophyte means living where it's wet. Halophyte or halophyte is living where it's dry. Hydrophile would be water loving, and hydrophobic would be water hating. Suggest and explain how the root hairs of halophytes are able to absorb water by osmosis from the soil of the salt marsh. Okay, well, we know that ions get actively transported into roots. Again, this is kind of GCSE, but a very tricky GCSE question. So, suggest and explain how they are able to absorb water. Well, all plants absorb water by osmosis by first actively transporting ions. Then water follows by osmosis. It's sort of drawn in by those ions, okay? So the root hairs will have lots of mitochondria, and transport proteins, for active transport of mineral ions, this will create a more negative water potential in the root hair than in the soil. So
water will follow by osmosis. Okay, so it's all about being able to set up that more negative water potential in the root hair. Okay, so it says suggest and explain, so it must have plenty of mitochondria to provide the energy for active transport, lots of transport proteins so that we can have that active transport across the membrane, set up the water potential so water will follow. Okay, next one. Even the smallest DNA molecule is very long. So this is Miss Dixon. The last one, I think, is me. Yeah, that's me. A kilobase, KB, is a unit equivalent to 1,000 base pairs of a DNA molecule. So I'm just going to put 1 KB equals 1,000. I'm just going to put BP for base pairs. 1 kilobase of double-stranded DNA has a length of 0.34 micrometers. So we can say that is also equal to that. Okay, so 1 kilobase, is equivalent to 1,000 base pairs, which is equivalent to 0.34 micrometers. Move that over a bit. The DNA in the nucleus of a cell from a fruit fly, is 5.6 centimeters long. Calculate the number of kilobases in the DNA of the fruit fly. Show your working and give your answer to the nearest whole number. Right, wants the answer in kilobases. So if it's 5.6 centimeters long, let's convert that into micrometers so that we can work with this, what we already know. So it's just a little puzzle to solve. Okay, so 5.6 centimeters is the same as 56 millimeters. And then we times it by 1,000 to get micrometers. 56,000 micrometers. Then if we do this number divided by this number, that'll tell us what we need to times this by. Okay, so 56,000 divided by 0.34. Oh, hold on, where have I got 0.34? Oh yeah, sorry, yeah. 56,000 divided by 0.34 is equal to 164,705.882 kilobases. I don't know what I was on about with that will tell us what to times it by, ignore that bit. Let's just double check. So yes, it wants it in kilobases. Did it say how many decimal places or significant figures? Give your answer to the nearest whole number. So 2 keeps that as an 8, 8 makes that a 9, so that makes that a 6. So we've got 164,706. Okay, next one. The DNA of the fruit fly was analysed and 22% of the bases were adenine. What percentage of the bases were guanine? Show your working. Okay, well it's DNA, so we don't need to worry about uracil. If 22% is adenine, then 22% must also be thymine. So A equals 22%. A always joins to T, so that must also be 22%. That means we have 56% left over. So the remaining DNA must be C and G, and they've got to be equal to each other because they join to each other. So half of 56 is 28. And then if we just want to check to make sure that our calculations make sense, 28 add 28 is going to be 40, 56. 56 add 44 is 60, yeah, it's 100. Yep, so we've got 100%, double check. 22% were adenine. How much was guanine? 22% adenine, yep, yep. So it wants to know this one, so 28. Okay, just have a drink. Okay. DNA molecule contains polynucleotide strands. Poly meaning many, nucleotides. Individual nucleotides are joined together to make polynucleotide strands. Ah, that's what I just said. How clever of me. What type of chemical reaction takes place when two nucleotides in a single polynucleotide strand are joined together? Well, you should know that water is given off when they join together. I can sort of visualize it now. Oh, this is still Mystic Sim. When water is given off, we call that a condensation reaction. Name the chemical released. Well, if it's a condensation reaction, then it's going to be water, isn't it? Or H2O. A DNA molecule contains two polynucleotide chains. Describe how these two chains are held together. Okay, so you need to think. So we have our ribose sugar, don't we? And then I can't remember exactly how it joins, so I'm just going to sort of trying to remember. Something like that. And then we have our base. Oh, I can't remember. Sorry, guys. It's something like that. And then our other bases. Then we've got our ribose sugars again. Which, sorry, that looks really bad. You've got your bases in the middle, okay, which are held together by hydrogen bonds. So bases are held together with hydrogen bonds. And then you could add in A to T, C to G. And then these phosphodiester bonds, which join the ribose sugars together, form what we call the backbone. So you'd end up with ribose, 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 ribose. And then we've got our bases in the middle, which are held together by hydrogen bonds. Okay, so hydrogen bonds down the middle, phosphodiester bonds hold the backbone together. Phosphodiester bonds join the sorry, I kept saying ribose, didn't I? Obviously that's RNA. Deoxyribose sugars, brain, together to form the backbone. Now, do we need to talk about the twisting? No, it just says describe how they're held together. I guess you could do, how many marks is it? 3 marks. You could do, actually, you could then say about the double helix structure for extra support. Let's see if it says it in the thingy. Phosphodiester bonds, hydrogen bonds, A to T, C to G. Reference to correct number of bonds between base pairs. 2 bonds between A and T, 3 bonds between C and G. So you could add that detail in like that. I think that's it. Yeah, it's not actually mentioned the double helix structure. Okay, so that's that one. So here we've got 1 mark for hydrogen bonds, 1 mark for saying A to T, C to G, 1 mark for saying 2 bonds there, 3 bonds there, 1 mark for saying phosphodiester bonds. How many marks? Oh, it's just one mark for phosphodiester bonds form the backbone. So yeah, we did good. Okay, so that's Miss Dixon again. Oh, I hope you can see all of that. Okay, next question 23 is my stuff. The Sumatran rhinoceros, Dicerorhinus sumatrensis, obviously, is a rare member of the family Rhinocerotidae. These rhinoceros are now critically endangered, with only 6 substantial populations in the wild. 4 in Sumatra, 1 in Borneo, and 1 in the Malay Peninsula. D. sumatrensis lives in rainforests. Their numbers are difficult to determine, but they are estimated to number fewer than 100. Complete the table below showing the classification of the Sumatran rhinoceros. So, here's our keep putting clothes on. Keep putting clothes on, fat, greasy slangs. C is class. Don't put the mnemonic in. Don't think the examiner would give you any marks for that. Class, order, genus. Now this is the trickier bit, so you get 1 mark for getting that right. This is the trickier bit. So kingdom, you need to think about what the kingdoms are. So we've got Animalia, Plantae, fungi, prokaryotes, protoctista. So it's going to be animal, isn't it? Animalia. Oops. Chordate means it's got a backbone. Species. Let's have a look back in the question. I believe it tells us D. sumatrensis. So capital D. You could write it out if you want. I don't think you'd lose any marks. Sumatrensis. Now it's important, those of you who don't have the best handwriting, it needs to be obvious that that's a capital and that that's a lowercase. Okay, and then underline it because it's hard to tell if it's in italics or not, so it's easier just to underline it. The name blah, 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 is part of the binomial naming system. State an advantage of using this system rather than using the common name Sumatran rhinoceros. I think the word universal is a really good word to use when it asks about the binomial naming system. So you can say it is used universally, or it's a universal language. So language won't cause a barrier to identification, or it won't cause any confusion, less confusion. That's the one I'd go with. I think the mark scheme says something else as well that you could have. Yeah, it's universal, it's recognised worldwide, no language barrier, we said that. We know which genus it belongs to as well as its species. Yeah, that's a good point, I didn't mention that. And different languages would have a different common name for the same organism. So yeah, any of those. I think saying that it's universal it's recognised worldwide is the safest sort of one to go with. Suggest 2 reasons why this species is critically endangered in the wild. Right, let's go back and see what information it gave us. The Sumatran rhinoceros is a rare member of the family Rhinocerotidae. These rhinoceros are now critically endangered, with only 6 substantial populations in the wild, 4 in Sumatra, 1 in Borneo, 1 in Malay Peninsula. D. sumatrensis lives in rainforests. Their numbers are difficult to determine, but they're estimated to be less than 100. So it lives in rainforests. Well, what do we know about rainforests when we've studied biodiversity? And GCSE, and probably all through high school and primary school. We know that the rainforest is being chopped down so that we can use the timber, so that we can clear land for agriculture and all sorts of things. So deforestation, I mean, it's a rhinoceros, so the first thing you might think, the first thing that came to my mind before I went back and read it, was that they would probably be hunted or poached for their horns, for the ivory. So I'd say that would be one. Hunting/poaching for their horns, for the ivory. And then I would, because it tells us in the question that it lives in the rainforest, deforestation or habitat disruption in the rainforest. Okay, so this is still my stuff. The remaining populations are all small and are scattered in isolated areas. These are factors that might speed up the extinction of the species. Explain why. Okay, so if the remaining populations are small, that means that the gene pool is going to be really, really limited. That means that they're going to be much more susceptible to a single disease wiping all of them or none of them. But generally, like look at the coronavirus now, if we all had the exact same genetics, every single person who got coronavirus would have died. If everybody had the same genes, which unfortunately some people have, which have resulted in their deaths from it, if we all had that, we'd all have died. But we have a mixture, obviously. So, small populations, small isolated populations mean small gene pool. So there will be low genetic diversity. This means that any geological or environmental change or disease could wipe out the whole population as they'd all have the same or very similar susceptibility. Captive breeding programs have been unsuccessful. Suggest one other way in which zoos can contribute to the conservation of the Sumatran rhinoceros. So they've not been able to capture them and get them to breed. How else can they help contribute to the conservation? Well, they can educate the people in the area so that people growing up can tell other people about it. They might be able to raise money to help fund further education and set up restrictions on areas, prevent poaching. The children that grow up knowing that information are less likely to poach and hunt if they know the situation and why it's bad and so on. So it's only 1 mark, so I think education's a good one to go for. Education, making locals aware, so less likely to hunt, poach them. You could have also said, what did I just say? Raising funds, setting up charities to raise money for what's it called? Protecting areas. Either of those is good. Is there anything else you could have said? Education, support for conservation projects and research. Yeah, pretty much. Okay. How are we doing? Whoa, that's a very long one. Okay, let's stop and do another one.